So we'll have to spend another 1 hour. Right?
It's like giving Nobel Prize to Obama in anticipation of what he's about to do.
Okay. Okay. Now.
And then he goes and drops phone bombs on all the.
So thanks for the introduction, and thanks Ranjini and Mahesh for putting up this very nice school and conference, and for the invitation. I'm going to talk about the correlation between static and dynamic properties in Confluent systems. So while preparing that talk, actually I realized that it was a bit overambitious, and so I'll talk about the dynamics part a bit less, and towards the end of the talk. First, I will go into the static part, and it will be somewhat complementary to what Rajesh was talking yesterday. The aim, the overall long-time goal in this direction, that what we want to understand is morphogenesis. And just in the previous talk, Madan was talking about morphogenesis. And it's a sort of evergreen problem in biological, in developmental biology. So pattern formation or symmetry breaking is very important, and at different stages of development, different type of patterns usually are formed. And the kind of problems that we want to understand is, for example, embryogenesis, patterning in Drosophila wing disc, organ formation in general, etc. So this type of problem. So now let's look at the basic ingredients of pattern formation. What you have is a morphogen gradient, and then you have a collective of cells. The morphogen gradient, it will have to interact with the collection of cells, and the cells will have to move. And also they will have to have different functions for this type of purposes. For example, they will have to divide, they will have to die, they will have to differentiate. Yeah. And so what we have is this dynamics part has been understood in a more better form. And it is known that the confluent cellular monolayer, they usually show glass-like behavior. And I'll get to it towards the end of the talk. First, I will discuss about the other aspect, which is the function aspect. So how can we describe function? Now, biological function is usually a complicated, is a complex problem, but it has been shown in last couple of decades and more that cell shape can be taken as a precursor of functions. So it has been shown that cell shape can affect crucial functions. Doesn't seem to, such as division, growth, apoptosis, division plane orientation, differentiation, etc., etc. Now, in human body, there are about 200 different types of cells of varying shape and functions. Here, I'm showing two different snapshots of human bronchial epithelial cell taken from this paper. So this is non-asthmatic and this is asthmatic. As you see, the cell shape in the asthmatic and non-asthmatic monolayer, they are quite different. Also, cell shape changes progressively during different processes such as cancer progression, asthma progression, wound healing, development, and many others. Furthermore, cells are functional units of tissue. So therefore, they should determine the tissue shape. So cell shape has quite important role in different kind of processes. Now, cellular functions, they depend on complex chemical signaling network. But here we are taking a mechanistic point of view where cell shape is related to the shape gene interaction network, which in turn determines the cellular function. The question is somewhat like in the statistical physics. Can we now treat this shape gene interaction network as a black box and take cell shape as a representative for the as a function for the cellular functions? So cell shape is the parameter that will control the cellular functions, and this is the point of view to in this direction. Now, this particular aspect, shape versus function, it has been explored in many different works, but they are mostly done either at the single cell level or at the level of average cell shape. So, for example, in this paper that appeared in 2011 in Cell, they took sea urchin cells and then they grow them on pattern substrate with different shapes. And depending on the shape, the division plane, orientation of the division plane as well as the division rate, they depend on the particular shape that they are growing the cells. In this particular paper, they have again investigated the similar aspect, the orientation, myosin plane orientation, but this is done at the average level, and so is in this one. So either it has been investigated at the single cell level or at the level of average cell shape. But as you see in this snapshot, again in that non-asthmatic monolayer, the cell-to-cell shape vary quite widely. So the question becomes, why should the average be enough, or is the shape variability too complex to understand? Does it have any importance, etc.? Now, in 2018, in a seminal work, the group of Jeff Friedberg, they showed a remarkable result. They showed that cell-to-cell shape variability across diverse epithelial monolayers, they follow a nearly universal distribution. This is the paper that appeared in Nature Physics. What they did, they look at the cell monolayer. For each of the cell, they write down the gyration tensor. And so this will be a 2 by 2 tensor. Now they diagonalize it. So if you diagonalize it, you will get 2 eigenvalues. These eigenvalues will have a dimension of length square. So they basically represent the major square of the major and minor axis of an equivalent ellipse. And if you take the ratio of those two major and the minor axis, you get the aspect ratio. And this is what they calculated. And this is the plot from the paper. If they plot the probability distribution function of the aspect ratio for different system, human bronchial epithelial cells, the MDCK cell, Drosophila cell monolayer, etc., they see some particular distribution. But when they plot this particular quantity x, which is the ratio of the aspect ratio minus 1 divided by the average aspect ratio minus 1, what they find is quite remarkable that for this diverse epithelial monolayer system, they seem to find very similar distribution for this particular quantity x. They fitted this distribution and these experimental data with this particular function, k gamma function, which is a single parameter function. If you notice, there is only one parameter k to the power k divided by gamma k, x k to the power, k minus 1, e to the power minus kx. When they fit it, they get the value of k. And what they find that for diverse epithelial system, they find a value of k which is very close to 2.5. Furthermore, there is another fascinating result in this work, which is the function standard deviation as a function of average aspect ratio. When they plot the standard deviation of this aspect ratio as a function of the average, they find that for different systems, the points follow nearly the same line. And moreover, as the system becomes more sluggish, as the dynamics become slower, with passing time, the points on this plot move towards the origin. So this is another experimental findings. Now let me first go through this rationale behind this applying k gamma distribution for this particular set of data. What it was shown earlier that the slow dynamics in this confluent epithelial system, it can be described by the dynamics of a vertex model. Vertex model I'll get to in a while what this model is. But within this model, there is a rigidity transition which is very similar to the jamming transition. Now, in a defined context, the tessellated volume in a jammed system follows a k gamma distribution. So, if you take each of the particle as the center and then take a Voronoi tessellation, the volume of those tessellated polygons, they follow a k gamma distribution. So, the authors ponder whether one can describe the shape distribution as well via the same function, which is a k gamma function. But there are a couple of points. First of all, this jamming transition, it is found within this particular model known as the vertex model. There are other models of confluent system as well, such as Voronoi model, cellular pots model, etc. And in those models, you do not have this rigidity transition or the jamming-like transition. Furthermore, biological systems are active. So what it means that they are mostly in the fluid regime. So therefore, why the jamming-like scenario should apply is not clear. More importantly, there is a particular aspect of volume that is used in the derivation of this k gamma distribution. So this aspect is that volume is an additive quantity, and this is crucial in the derivation. On the other hand, aspect ratio is not. So therefore, why this particular distribution should apply for the aspect ratio is not clear. In fact, the authors wrote, and I'm paraphrasing from this paper, there currently exists no rigorous basis for the applicability of the k gamma distribution to metrics of cell shape. Yet, in the later experiments and simulations, several works have shown that not only you can apply this k gamma distribution to this distribution of cell shape or the aspect ratio, when you apply this, you get a value which is very close to 2.5. So here I am showing data for cancer cell lines, all of them. These are data from different numerical simulations. The abiotic system, these are data for active vertex model, etc., etc. And if you fit this k gamma function to this distribution, you get a value of k which is very close to 2.5. Although there is a slight deviation, you actually get from 2 to 4, something like that. So then we wondered whether, since these are very generic results, there must be some more fundamental basic reason behind this observation. Huh. Yeah.
Is there also a volume distribution from cell to cell, or that volume is fixed?
Yes, there is a volume distribution as well, and thought of skipping that in the interest of time. The volume distribution is actually the K gamma distribution. Sorry,
The volume distribution.
is actually the K gamma distribution. This one was for the volume distribution, and they applied it for the cellular shape. But for the volume distribution, you can actually derive that it will be a K gamma distribution. No, K change in that case. I mean, K can vary. It has a large variation, in fact. Okay. So in fact, K can be very as large as like 50 or 100. So it can be really sharply peaked. Okay, so the questions that we tried to, so AI is very close to Gaussian in that case, but it can be very small as well. I mean, there's a huge range. So the questions that we wanted to address in this direction is that first, what is the origin of this universality? What sets this specific value of K 2.5? And another question, whether these findings are truly universal, because if something you want to address, whether it's universal or not, doing it from experimental or simulation data is non-trivial. So therefore, this and what are the consequences? So answers to these questions require an analytical description, and this is what I will first do, and then we'll get to the comparison. Can you repeat?
Also, the shape index shows this similar kind of gamma distribution with K equal to 2.5.
So there are a couple of points. The K gamma distribution that they actually fitted is not to the shape, not to the aspect ratio, but to the scaled aspect ratio. Now, for the scaled aspect ratio, if you fit with one particular parameter, that is not really universal, right? I mean, if it is universal, you must have K same value. But if K varies, then it already gives you the impression that it cannot be universal. So I'll get to that, okay? But it was mostly fitted with the K gamma function for the scale aspect ratio. Okay, so first let's go through a simplified physical description of cell. So for most practical purpose, the mechanical properties of a cell is given by a thin layer of cytoplasm, the actomyosin, thin layer of cytoplasm made of actomyosin, which is known as the cell cortex. And then there are different adhesion molecules for cell-cell interaction, and also the cells can interact with the surface, which is via the focal adhesion and something like that. Now, one particular aspect of these monolayers that here these are confluent monolayers. So that means the packing fraction is always unity. It is not a control parameter. You cannot change it. Now, another experimentally, it is seen that the height of the cells remains almost constant at a particular time. So therefore, one can actually describe this system as a two-dimensional description. So these are some figures taken from this paper. So now one can describe this system via this energy function.
So this height remaining constant, is it universal for all the cell types?
It fluctuates a little bit. So this is what
for
this.
all cases, like because you showed wide, very wide variation of cell types.
Yes.
That follow this case.
Yeah, so these are the type of system that we are interested in this one. But if you do not have confluency, then it can vary quite a lot. But here we are interested in the confluent system. There, for different system, this property holds. So if it is varying a lot, then this framework will not work. So as a function of time, it varies a lot, but within a particular time, yeah. So those systems we cannot explain by this type of framework. So this framework applies when the height is not varying too much. So now when the cell cytoplasm, this can be represented as an incompressible fluid. So the volume remains constant and the height fluctuates a little bit. So those two aspects we can write down in this area constant, AI minus A naught square. So AI is the area of the particular ith cell and A naught is the target area. So if height does not vary too much, you can actually represent it as a constant like this. Now this second term, this is the.
So is it too difficult to model this cylinders like that, where you can also allow this height to fluctuate?
You are allowing the fluctuation, right?
No, but you are saying that the height cannot change. So you are
it fluctuates less. If it fluctuates too much, then this description does not
No,
work.
but I'm
well.
saying that this vertex model, if you also include this height
Hmm.
and another term in this Hamiltonian, so will that be a very wrong thing to do?
It is included. It is included via this constraint, right? If your volume remains constant, then your height variation you can actually represent via the area. So A naught need not be constant. A naught can vary.
No, but I'm saying that you put both a constraint on the top layer
H and A.
and H. So H you can also put another harmonic potential.
You do not need it. So if the volume remains constant, your variation in H can only come via area.
No, but you're saying that there are certain occasions where you can have that fluctuation.
No, so then the complication is different. That if it is strongly fluctuating, this framework will not be good. But yeah, you can include it in some form. This framework will not be good in that case.
Okay.
Okay, so
But
this next is the cell.
Here you can incorporate this polydispersity in A0, right? So that can
Yeah,
take
exactly.
the
Yeah. So I didn't write it because it will look too complicated, but A naught you can specify for each of the cells. And there are simulation where it has been done. Okay, so this is the cell perimeter term. What it does, there are actually two terms, this lambda p times pi square, and then there is proportional to pi. And there is another part which is constant, you can actually ignore. So this first part, it is mostly encoding the property of the cellular cortex. And the second part is encoding mostly the property of these adhesion molecules. So cell-cell adhesion, cell-cell interaction part. So this, since interaction is only taking place at the cell perimeter, therefore it must be proportional to pi. And these two terms one can combine and write it as a perimeter constant P naught. So P naught, although one calls it as a target perimeter, it actually encodes the cellular cell-cell interaction. Okay, another point, so once given this energy function, now one needs a particular temperature to define the dynamics. Now this temperature is a non-trivial quantity here because it includes all possible activity as well as equilibrium temperature. And I will get to it in a while, in what scenario one can include.
And perimeters should have some other prefactor.
They actually are different. So here, for example, that prefactor that you are talking about, lambda p times p naught. So you need to have two parameters and you have it, lambda p and
Yeah,
p
but
naught.
P naught is kind of fixed, right? Because it is some initial.
No, p naught is a parameter. Pi is the perimeter, but P naught is a parameter. You can vary it.
Okay.
And depending on P naught, actually the property changes quite.
For a given system, P naught is fixed, right?
No, it's a condition. It's a parameter. You can vary it. Depending on whether you do some RAB5 or RAB12 or some other drug, it can change quite drastically.
Okay.
Okay, so now given this energy function and the temperature, now you need models of confluent system. So what that means that you need models where the cells entirely cover the space, and these models usually represent these the cells as polygons. And there are different types of model. There are different continuum model known as the vertex model, Voronoi model, and then there are discrete lattice-based model known as cellular pots model. And you can actually simulate it on different type of model like squared lattice or hexagonal lattice, etc. And then there is a combination of these two, which is known as the phase field model. In this work, we used both cellular pots model and the vertex model. So one discrete model, one continuum model to test and verify our predictions. And there are some snapshots shown from our simulations. So this is for vertex model, this is for Voronoi model, and this is for cellular pots model. So the difference is basically the vertex model, the degrees of freedom are the vertices of these polygons. In the Voronoi model, it is the center of mass. So these Voronoi centers are the not center of mass, Voronoi centers are the degrees of freedom, and here it's a lattice-based model, so it has more microscopic structure, cellular pots model.
Nobel constraint is only on the area.
Only on the area, yeah. So now given these models, the energy function and temperature and these models, our aim is to write down a analytical description for the aspect ratio. So what we followed this prescription given by this, followed by this experimental paper, we also write down the gyration tensor, we diagonalize it, we get the two eigenvalues and which will be square of the major and the minor axis, and taking the ratio of those two axes, we will get the aspect ratio. Now, a direct calculation is somewhat challenging, so we took a slightly indirect route. What we do, first calculate the distribution of the squared radius of gyration around the center of mass. So the reason it's slightly easier that S1 and S2, let's say, the major and the minor axis, those are anisotropic quantity. So getting their distribution is slightly non-trivial, and this one is a isotopic quantity. But given this distribution for the squared radius of gyration, we can actually get the aspect ratio using some relations. What we do is represent the cell via a set of points on the perimeter, something like this, represented by the vector x. And then one can formally write down the distribution for the squared radius of gyration, which is, so this is the, this I'll get in a while. So this is some distribution where the configuration, this is assumption that the configuration is appearing as a Boltzmann-like distribution. So it's not exactly Boltzmann because the temperature has a different notion, but we'll get to it in a while. This delta function, this second delta function picks out specific component of S squared, and the first delta function comes from definition that the center of mass of the cell must coincide with the center of the coordinate system. And Z is partition function. This is a complicated way of writing the volume element, and so we are actually following the notation from polymer physics literature. Okay, so I'll not go into the detailed analytical derivation because that will take time. Instead, I'll just point out the main points or what is the, how to obtain the derivation. So this is somewhat of this type. Consider that you know the distribution P of x of a particular quantity x. If you know that and you want the distribution for y, which is a function of x, one can actually obtain the distribution performing this delta function y minus f of x. Weighted with P of x, all possible x, and then integrated over all possible x. So this delta function picks out the specific component of y from the x, and then you get the distribution of P of y. So this is somewhat similar. This, if we consider this distribution, this delta function picks out those specific component. This actually comes from definition, and this is what we'll be following. Now, this is quite formal and general, but coming from here to somewhat more useful requires several approximations in the analytical derivation. These approximations are either motivated or tested in our simulation, and this is what I will show in a while. Now, these are the S1 and S2 are the major and the minor axes, if we call them, and S, which is the radius of gyration around the center of mass, they are related via this S1 square plus S2 square is S square, and this gives you the area A. So here I have not written the constant pi and 2. But from here I can write down S square by A as using this relation R plus 1 over R. So in the derivation you will get this particular combination R plus 1 over R as you go into the final form. Yeah.
For a polygon, how are you defining major axis, minor axis? Like it
So,
can be of any orientation.
yeah, so
right?
it's actually not the major axis and minor axis. What we are doing, write down the gyration tensor.
Okay.
So from the gyration tensor, you
Whichever
diagonalize, and the two eigenvalues give you the major and the minor axis.
Whichever
Which one is
greater,
larger,
yeah,
it gives you the
I see.
major axis. Yeah. Okay, so now let's look at the approximations, and what we want to do is write down the key ingredients within the theory, and the key ingredients here are kept as the energy function, which combines these two, the constant, area constant and the perimeter constant, and then various confluent models, and then the effective temperature T, which will include activity as well as equilibrium temperature. But apart from them, there are several other aspects as well. The systems will have, for example, cell division and apoptosis. They will have cell propulsion, etc. Now, first let's consider the cell division and apoptosis. Now, within this system of our interest, the cell division and apoptosis, their number becomes really rare as you go towards the confluent system. So, starting from say very few number of cells in the dish, then they will keep dividing, and some of them will die. Eventually, they will reach the limit where all the cells entirely fill the space. As you go towards that confluent limit, the cell division and apoptosis rate keeps decreasing. So, for example, some experimental number in the MDCK monolayer and taken from this paper, cell division is of the order of 10⁻² per hour per cell, and this is before the confluency limit. So, as you go towards the confluent limit, this number becomes even smaller and smaller. On the other hand, apoptosis, so this is actually post-confluency, this is the number we could find, this is of the order of 10⁻² per day per cell. And as it approaches the homeostatic, the steady state, then these numbers become even much more less. And it was shown in many works that these processes can be ignored for the glass-like dynamics or the wound healing type of scenario. So this, for example, you can find a discussion on this paper. In the theory, we have neglected them in the analytical derivation, but we did keep them in the simulation just for checking. And if you keep them in these rates, division and apoptosis, and if their rates are not too large, then the theoretical predictions still hold. So this one can show. Now, what about the self-propulsion? So self-propulsion is usually important, but in the first version, we did not keep it. The motivation was the following, that without the self-propulsion as well, there are many processes that one can understand. For example, cell sorting, chemotaxis, this glass-like dynamics, statistics in the confluent monolayer, etc., etc. In this type of theoretical derivation, you don't need the self-propulsion to understand the experimental scenario. But more importantly, as also Sanjeev was talking about, that if the tau p is very small, the system becomes more like, the active noise becomes more like the passive system. This one can also show, and this is from our own paper, within the mode coupling theory, you can analytically show that as a function of different tau p, if you look at the long time behavior of the system, the T effective at very short time, it's the bulk temperature, but at very long time, it approaches a defined value. So it's defined from the equilibrium temperature, but it approaches a constant. And this is another result from Bandyopadhyay and Kurchan's paper that appeared in Nature Physics. So the slope gives you the constant effective temperature. So the interpretation of T is non-trivial, but if the system, within these models, the status of this temperature has a equilibrium-like temperature. If that is there, then one would expect that the distribution of the configuration will follow a Boltzmann-like distribution. And this is what we tested. So we are plotting the energy as a function of log of PE. Now, if it is Boltzmann-like, then you will see that it is a straight line. Now, apart from the first couple of points, it seems to be agreeing. And moreover, if it follows this distribution and if you plot this scaled by the temperature T, E over T, then it should fall on a master curve, and it also falls on a master curve. Another couple of points. The first is that this constraint of confluency. Now, this is a very tough mathematical challenging problem, including the constraint of confluency, and not much known analytically. But what we showed that for the distribution of the aspect ratio, distribution of energy. So what we did, we are simulating at different time, we are just calculating the energy using this energy function, and that follows this distribution. The
Gaussian distribution around the average with CV giving the fluctuation. That's the dynamical distribution.
no, energy it minus. So this I think this p factor that we did not include, but this will be distribution will be e to the power minus e.
Distribution of state is e to the power minus e.
So what we are doing, we are looking at the states that are allowed given this energy. What is the probability? So that should follow e to the power minus e. Okay, maybe we can discuss later. We are just okay. So, so the other aspect that the constant of confluency, and doing this calculation is analytically is very, very hard. What we find that actually for the
see, your Hamiltonian has the area and the perimeter.
Yeah.
And this shape aspect ratio, you, I mean, that information was initially not there, right? So
Not
you
there,
build
not yet.
it in between somehow, you compute the,
we will compute that. I mean,
from the equilibrium configuration.
so for each of the configuration, we'll from the configuration we'll write down the gyration tensor. We'll diagonalize it, take the ratio of the square root of the eigenvalues, and that will be the aspect ratio. So till now I haven't shown that relation. Now the first point is that if the constant of confluency, so what I'm going to argue and that the constant of confluency is not crucial for the aspect ratio distribution. So we have argued in several ways. Some of them are as follows. If the constant of confluency is crucial, then the aspect ratio distribution for a single cell and for a confluent monolayer, they should be different if you keep all the other parameters same. So we simulated a single cell in a medium and a confluent system, keeping all the other parameters same. And the points are for the distribution of the single cell, and the lines are the distribution of the confluent system. So what it shows that the constant of confluency is probably not very crucial for the distribution of the aspect ratio. Also, aspect ratio is a dimensionless variable, so one would expect that maybe the area is not that crucial. Second, what we see, and this is so this mu, we actually have a derivation. This is the PDF of the area distribution, and the mu can be really, really large. So it's a very sharply peaked function. But more importantly, when we change this lambda a, if this term is really crucial, we should get that the distribution of the aspect ratio should vary with varying a. What we find is something quite different. As we vary lambda a, the distribution of the aspect ratio does not change at all. They fall on top of each other. And also, this, if you were in the talk by Rajesh yesterday, that actually this area constant is not crucial for the aspect ratio, as he also showed. So then one can ignore this area term and write down and go through the derivation to obtain the aspect distribution of the aspect ratio for the distribution of the aspect ratio R. So here aspect ratio I am writing as a R. And one obtains this particular form where n actually is a normalization coefficient, and we have the exact expression for that. So this is not in parameter. R plus 1 over R, so this appears because of that particular combination that I was showing earlier. This to the power 3 by 2 times 1 minus 1 over R square e to the power minus alpha R plus 1 over R. So this 3 by 2, it comes for a particular mathematical property. So for a closed loop object, you will always get it 3 by 2, and it's not coming from any other details. All the system specific details, they enter into a single parameter alpha, and this is written here, lambda p times 1 minus k p naught divided by t. Or p naught was that parameter, lambda p again this lambda p of the perimeter constant, k is a constant, and t is the temperature. So what happens that all the system specific parameters, they combine into a single parameter, and that determines the cell shape variability. And this has important consequences for the universal properties. This is the area distribution. So this again, I mean, here the constant of confluency is actually crucial, and we included in a phenomenological way. This is just to show that one can obtain this distribution, and this is same as that is obtained for the Voronoi area volume or area distribution. And it provides an alternative way to think about the constant of confluency. We haven't done much yet, but one would like to do something in that direction as well. But more importantly, in this context, mu and alpha, if one know together, they can actually distinguish the effects of changing different parameters within the system, within the experimental system.
Saroj?
Okay.
So you compared the situation between a single suspended cell and a cell in a confluent monolayer.
Yeah.
The third scenario is when the monolayer is not confluent.
Yeah.
Wouldn't you consider that scenario, the third scenario, where you have cells on one side, but there is a gap on another side, to be more asymmetric as compared to?
So you cannot have gaps, you need a medium, but we did it actually. I mean, several cells in the medium. So that also gives the same result. And that you can do only in the cellular POTS model, not in the other models. So now let's get into the results. If I plot the distribution of R for different value of alpha, they looks like this. So for different value of alpha, alpha determines all the system specific parameters. Now, when one plots this particular quantity, scale aspect ratio as they did in the experiment, which is the ratio of the aspect ratio minus 1 divided by the average aspect ratio minus 1, one sees that the distribution looks like this. So the dependence on alpha becomes really, really weak when one plots the PDF of the scaled aspect ratio, and they are the same values of alpha as is there. And this is, so what it shows that the alpha dependence is there, it is weak, but it's not strictly universal, the distribution. Another aspect, if one ignores 1 over R here compared to R, then if you obtain the PDF of the scaled aspect ratio, you will get strictly this k gamma function. So that's why when one fits the k gamma function, it seems to work somewhat, and one will get in that case k to be 2.5. So that 2.5 comes because of this particular quantity 3 by 2. So if this is 3 by 2, this in the K-gamma distribution representation, it will have to be 2.5. So it comes from this geometric property. But this is not a good approximation because R is of the order of 1, and therefore one will not get this particular distribution. So there is a variation when one feeds the experimental data, there is a variation of K. It comes from 2 to 4 or something like that. Now, there is another aspect which is actually universal, and this aspect comes because of the fact that all system-specific parameters gets clubbed into one particular aspect, one particular parameter alpha. So when one gets standard deviation and average aspect ratio, so I can formally write as some function, the standard deviation as a function of alpha, average aspect ratio as another function of alpha. Now I can eliminate alpha from these two equations, and this relation, standard deviation as a function of average aspect ratio, it becomes independent of any system-specific parameters. So therefore, it must be same for all systems. And this is what it looks like if I plot from the analytical expression. So actual expression is actually quite complicated, but it looks nearly linear. So if I fit a linear expression, it looks like 0.71 R minus 0.75. So now we tested these results in our simulations. We simulated both the cellular POTS model and vertex model. And in the cellular POTS model, I'm showing one particular parameter that we are changing, lambda p. Here I'm changing temperature, but we have also other parameters which we tested. The PDF looks like this. The points are simulation data. The lines are this expression that we obtained. Now, when we plot this scale aspect ratio, as we showed in the analytical result as well, they nearly overlap on top of each other. So this is the near universality aspect of the theory. The other aspect, the universal aspect, standard deviation as a function of the average aspect ratio. So here there is no fitting parameter, and the line is just the plot of the theory. The points are the simulation data, and they agree quite remarkably. So this is not a fit, this is just the comparison between the theory and the simulation data. Now this aspect that if you have both this mu and mu of this distribution of the area given by the mu and this alpha which governs the distribution of the aspect ratio, and if you plot alpha as a function of mu by varying only one particular parameter, so it's a bit clumsy plot, but let's go through slowly. So if you vary only lambda p, you see only alpha changes, mu remains constant. So lambda p actually is the coefficient of this one. If you vary lambda a only in the simulation, then mu varies, so which means the area distribution varies, but not the not alpha, which means the aspect ratio distribution does not vary. And when you vary t, both of them vary. So what it shows that lambda a and lambda p have distinct feature when you look at in terms of the distribution of aspect ratio and area.
So
Now, yeah.
what was your P0 in those previous cases?
P0 we kept fixed because we didn't know how to actually compare P0 or measure in the
Like
experiment. We kept it fixed 25 for this set of
25, like
25.
non
25 actually represents just below this rigidity transition or the jamming transition in the vertex
yeah,
model.
that's what, like which side it is, like
lower side. It's lower than 3.81.
Lower than 3.1, just solid.
Yeah.
Okay.
But we also varied P0, and so let me not get into that. Okay, so now let's look at the comparison with experiments. And here the experiment, I'm taking the data from this Nature Physics paper, which motivated this work actually. And here I'm taking the data from in the past MDCK cells. These symbols are the data from the experiment, and the lines are our theoretical fit. From the fit, I get alpha. Knowing alpha, you will able to calculate the PDF of the scaled aspect ratio. And this is just the comparison. So this is data taken from here. And this line is our this theoretical curve. So this we have done for all possible set of data, and some of them are shown here. The other aspect that if you know alpha, you can also look at the standard deviation as a function of the average aspect ratio. And as we see, similar interpretation was given in the experimental paper as well. As alpha increases, so with increasing time, like in this day, several days, these points actually move towards the origin. Now let's look at one of these near universality prediction of this system. So here I'm showing the PDF of the scaled aspect ratio for two different values of alpha, 2 and 6. And then I'll keep plotting the experimental data taken from different papers. So from these 4 papers. And as I keep plotting them in the same figure, so they should fall on top of each other. And this is what exactly we see. So note that the variety of the cell types here is truly remarkable. So there you have various type of cancer cell lines. There are asthmatic, non-asthmatic HBC cells. There are MDCK, Drosophila wing disc, simulation on active equilibrium vertex model, active Voronoi model, cellular POTS model, etc., etc. Yet all of them fall nearly on top of each other and consistent with the theoretical predictions. Now, the other aspect, the universal aspect of this theory, that if you plot the standard deviation as a function of the average aspect ratio, that should simply match. I mean, because there is no fitting parameter, and we have taken data from these three papers. And when we plot them, this line is the theoretical prediction, and we simply plot standard deviation as a function of the average aspect ratio, and it also agrees quite well. Now, let me get into one predictive power of the theory. So when you have only one particular parameter, when you have only one particular parameter, you can actually predict from the average the distribution. So average is a much less information, but you can actually predict the distribution from the knowledge of the average. And this is what we did. From this paper, we obtained the value of the average aspect ratio, and then we analytically calculated alpha. Knowing alpha, we calculate P of R, and then they are the plot of this distribution. And we did not have this data. So what we did, but we had this data of the scale aspect ratio. So we transform them into scale aspect ratio distribution, and then this again agrees quite well. Now, in the next few minutes, let me get into this dynamics aspect. So one of the results that was shown in the experiment, that as the system becomes more sluggish, as the dynamics becomes slower and slower, the data point on this plot, standard deviation as a function of the average aspect ratio, moves towards the origin. So we again explored this aspect in our simulation. We are plotting here the relaxation time as a function of alpha, which is this parameter that we obtained, and the average aspect ratio. So as the average aspect ratio becomes smaller, so as you go towards this direction, the dynamics becomes larger and larger, and this also increases with alpha. Now we had this particular relation that alpha should be proportional to 1 over temperature. So if I plot relaxation time as a function of T, so it looks like this. But if I plot it in terms of 1 over alpha, if I plot them on the same curve, actually it looks nearly indistinguishable. So this aspect now, this is actually, so now why cell shape and glassy dynamics, this correlation is so non-trivial. If you look at this system, they have a non-trivial feature. These systems are quite confluent at all times. So packing fraction is not a parameter anymore. Unlike particulate models, these systems are extended object, so a bit difficult to handle. And moreover, the average cell shape, which is a static property, that seems to control the glassy dynamics. And this you can compute and calculate in experiment. So a quantity that you can just measure from the static snapshot seems to determine what is going to be the dynamics. And these are some snapshots taken from, again, those simulations. So the non-trivial aspect, this is non-trivial because if you don't know about the glassy dynamics, it is defined as follows, that the extreme dynamical slowdown without much change in the static properties. So if you look at two snapshots of glass and a liquid, they look nearly identical. But here it seems that you can actually, from the static property, you can measure something and you can determine the dynamics from there. So in the Confluent system, the dynamics and the cell shape, which is static property, seems to be quite strongly correlated. Now, there is one particular theory that actually sort of advocates this type of scenario, and this is known as the mode coupling theory. Now, mode coupling theory is sort of, it has a strange status. It just seems to fail left, right, center, but it also seems to predict something which seems to be quite remarkable. So we thought that we'll apply this theory for this Confluent system, and we'll see how the theory fares. And this is what we did. So we take the mode coupling theory. I'm not going to the detail in the interest of time, but the main aspect is that you have the theory, and the theory says that if you know the statics, you can predict the dynamics. So in the two-dimensional system, from simulation, we can measure the statics, so which is the static structure factor or the G of R, something like that. And then we can feed that information into the mode coupling theory, and the theory will give the dynamics. Now, from the simulation, we can also calculate the dynamics, and then we can compare between the mode coupling theory and the simulation data. So we tested this aspect within the vertex model simulation, and one can find this in this reference. So this is some of these calculations. So what we did, take the snapshot, and from there we represent each of the cell by their center of mass. And from there, I can calculate now the G of R. So just like a particular system now, because they are represented by their center of mass. And from there, I can calculate the G of R, which is defined here, and the static structure factor S of K. So G of R looks like this. As temperature decreases, the peak height slowly increases. And here I'm showing 2 S of K. So this is with decreasing temperature, and this is with changing P naught. And now we are going to feed this information to the mode coupling theory, and we get the dynamics from the theory. We already have the dynamics in the simulation. We'll show the comparison. So this is what we get when we give the static structure factor to the mode coupling theory. Then this is the 2-point correlation function, fkt. So k max basically at the maxima S of k, where the fast peak. So that corresponds to this k max as a function of t, and this looks like this. And in the simulation data, q of t, it looks like this. And now, one of the prediction of mode coupling theory is the time temperature superposition. What it says that if you scale t by the relaxation time tau, or relaxation time you can define where the 2-point correlation function goes some certain value, say 0.3. So in that case, you will see a data collapse in the alpha regime. So alpha regime means the long time part. And we see the similar thing both in the mode coupling theory data as well as the simulation data. Now, this is a very strong test of the theory. So what mode coupling theory does, it says that there is a divergence of relaxation time at a particular temperature known as the TMCT. It is well known that this divergence is not there in simulation or experiment. Yet, in a particular regime of temperature, one can fit the mode coupling theory prediction, which is a power law divergence, and one sees that this relation seems to work quite well within a certain regime. So here we have done this exercise that feed the static data to the mode coupling theory, then from there get the relaxation time, and then represent it as a log scale t minus tmct, and the power law will be t minus tmct to the power 1.5 minus 1.5. So this shows a divergence at TMCT. When we use the same form in the simulation data, the divergence point is different, and that is because of some other reason. Let's not get into that. But if you use those TC and represent that T as a function of T minus TC, you get a power law, and the exponent is same. So 1.5. This happens in the regime, as Shantanu was asking, with P naught. If you go to the very large P naught, there is a result known as the Habermas regime in the large P naught regime. In that regime, these two exponents seems to agree quite well. But if you go to low P naught regime, then this becomes super Arrhenius, much like the point particle system. And there this exponent actually seems, they don't agree anymore. So in the Habermas regime, the theory seems to work quite well. This is another way of showing the comparison via the angel plot presentation, and this is tau as a function of Tg over T. If you look for the particulate system, and from Saroj or Andrea's talk, you see that it goes like this in this lower part. So this is known as the super Arrhenius relaxation. The state line is the Arrhenius, but you usually don't see in the other side, except for some very specific system, soft sphere type of system. In this system, you readily see Habermas behavior. And in this regime, if you fit the RFOT expression, it actually gives a negative TK. So RFOT does not seem to work in this regime, while mode coupling theory seems to work remarkably well. It is built in some sense for this regime. So, okay, so the agreement shows that the dynamics and the statics are also quite strongly correlated, and one can apply the mode coupling theory formalism, which is this mechanism that structure gives the dynamics and the dynamics is related to the structure. Just 1 minute. Okay, I'll go ahead.
No, this, as you said, that this harmonic sphere or Hertzian sphere at low densities, you can actually get Habermas.
Yeah.
Did you check MCT predictions on that equilibrium context?
I didn't, but Jamel did. Jamel and Bhartiya again.
But with the mode coupling?
Mode coupling theory, well, I mean, there actually you don't get a divergence in the simulation data. So this seems to be different from the Hertzian sphere data. Here it seems to give a divergence. So there it goes to a constant.
So it's not the Habermas regime which matters, right? Or is something else?
Yeah, I don't know what it is, to be honest. I mean, MCT seems to work. That is till now that.
No,
That's
but
the
MCT
result.
seems to work only in this vertex model type scenario, not in the harmonic sphere in the even in the sub-RNES regime.
Well, I mean, there it seems to not working because it always predicts a divergence, right? But at
No,
the
no, no.
very small. Go ahead.
So I'm asking this question: this if you take a Hertzian sphere, then there are the certain densities below jamming density.
Below
you get,
jamming density, yeah.
sub-RNES
Yeah.
behavior of the relaxation time.
Yeah.
Will mode coupling theory be a better predictor there as well, or?
So better predictor, I don't know. Maybe better than RF40, but they are strictly speaking, it's again breaking down. And that's what the result that they are claiming. Yeah. Here also, it doesn't show the divergence. I mean, it actually, so if you look at this, it doesn't diverge. I mean, the simulation data TC, if you go to lower and lower temperature, it will keep changing. But within the regime, it seems to have some value. And the strong point is that exponent is same. Okay, so let me wrap up quickly. Why these results are important? As I stated that we want to couple mechanics and the pattern formation. And the way that we are looking at this cell shape and functions, if they are very strongly related, and it has been shown that they are related at the single cell or at the average level. The question we are asking that are they also related at the statistical level? And so cell shape also governs this monolayer dynamics. So this is another aspect that the same parameter, it governs both statics and dynamics. Now can we use it in the morphogenesis type of framework? We are testing some of the aspects in experiments with colleagues from TIFR Hyderabad, and eventually we would like to develop this morphogenesis like framework within these epithelial and confluent monolayers. So just a couple of minutes, with Tomal we are looking at this self-propulsion part. So now we have included in the theory and the simulation, but we are testing some of these aspects and the predictions in the experiments. With Monish, we are looking at this particular aspect that can we use the cell shape as a precursor of division. So in Drosophila wing disc, we are, so my student Sovik, he is doing the experiments, and there we would like to see if we can find a relation that can be used as a precursor of the cell division. This last part about the dynamics, again, now we are doing the experiment as well, and with these people are doing the experiment, and we are trying to analyze the data. So we are exploring this relation of static structure versus dynamics in this confluent system. So we have a couple of data points, but it seems to be the right direction, but we hope we'll have some more data in near future. So to conclude, the distribution of the scale aspect ratio is nearly but not exactly universal. The standard deviation versus average aspect ratio is universal. Cell shape may be used for both static and dynamic properties, and we need to understand this in more detail. And one can get more detail about these aspects in these two papers. So this is preprint and it appeared in eLive. And thank you. So mode coupling theory, you can actually include a property of a particular system in two different ways. Modify the dynamic equations, or you take the other approach that the statics is known, everything is in the statics, you feed that into the dynamic, into the theory.
For the density.
Yeah.
Centroid density.
Yes. So it's.
You don't have anything to do. You don't have any.
So what
describing the shape?
Yeah, so this is actually a bit surprising that if you what we are doing representing the cell by their centers, and from there we are getting the static structure factor, and that we are feeding to the MCT, and even then it's, I mean, there is no.
Should it work?
Yeah, exactly. I was a bit surprised, to be honest, seeing this. But it seems that you have this information of cell shape in the static structure factor and G of R as well in some form that we need to understand in more detail. Yeah, for Voronoi model, yes. So now we are actually testing this. So there it's more straightforward.
So in both Voronoi and vertex model, the edge between two cells is basically flat. But in reality, it is not flat. It depends upon the pressure difference. Since again the Hamiltonian itself contains the perimeter term, which is the major part of the of your Hamiltonian.
Mm-hmm.
So the fluctuation should.
No, this is one of the reasons that I like cellular POTS model, which has some other difficulties. Within vertex and Voronoi model, it will have to be a straight line or with some constant curvature. Beyond that, doing anything is very, very hard. In cellular POTS model, your boundary can be anything; it can fluctuate, but then this underlying lattice structure will come into play, and
Maybe
so
the
this is
lattices
difficult.
can be like made very small. Then in that case, the.
So I know Max and his student, Max B and his student tried something with some varying boundary curvature, but in general, it's doing it's very hard, and it's not known how to actually do that. So it's yeah.
Continuous function.
Many more.
So let's say you have many small straight lines, which you put it on your vertex model. Because vertex model is defined by how many vertex you have. Each cell.
Yeah,
That
you
number
can.
If you increase, then
you can start with that, but once it goes to the ground state is a hexagonal structure. So you will get variation around that. And in experiment, you see that, I don't know if I had some plot, but in experiment, you will see some distribution, say, if, some 5, 6, or 4, 5, 6, 7, 9, etc., 8, 9, etc. So you will see that around 6 it will peak and it will decay.
But still you have a broad distribution. So even if think in the minimum energy configuration in this Hamiltonian, you still get roughly a hexagonal structure.
Yeah,
with some boundary.
Yeah. Yeah, in
should be
fact,
okay.
yeah, it looks very close to hexagonal, both in cellular POTS model and in this one as well. But you can have more fluctuation depending on the parameters.
Saroj, so within this model, your cells are like polygons, right? So, like, I mean, how precisely? I mean, how exactly you are defining the aspect ratio for this?
So the so one is the cellular POTS model or in the vertex model as well. So let's say you have something like this. So then you can write down, you can take this shape and say describe it as a set of points on the perimeter. Now knowing the set of points, you can write down the gyration tensor Ixx, Ixy, Iyx, Iyy. So and from there you can diagonalize. And you get the 2 eigenvalues and take their square root of their ratio, and that gives you.
Any more questions? Someone.
Well, if you go to very high temperature, you can get a very funny looking shape. But in the probably experimentally relevant regime, they are mostly convex. But it can be quite fractal-like structure as well, particularly in the cellular POTS model.
Okay, so let's thank the speaker today. Let's thank all the speakers of today's session.